Hello children, in today's lecture we are going to discuss about this acids, bases and salts. So we are going to continue with the lecture. So last lecture we came to know how we identify the acids and how we identify the bases and how we identify the salts, right? Now here one question is given. So here we are provided with 3 test tubes. One of them contains distilled water and the other 2 contain acid solution and a base solution respectively. If you are given only red litmus paper, how you identify the contents of each test tube? So that is the question given here. So here we know with the red litmus paper, right? So it is actually indicator. So if it is an acid solution, red litmus paper remains red in an acid solution. If it is a acid, there is no change in color. If it is a base, so red litmus will turn into blue. Red will be turned into blue in a basic solution. If it is a distilled water, so it is a neutral in nature. So red litmus paper remains red and no color change. So how we identify? Here I am giving you some steps to here. So we follow a particular procedure. So stepwise we can see. So first we take a red litmus strip. 3 test tubes are there. So first we dip it into the first test tube solution. So immediately if it turns into blue, we have to identify that it is a base solution, right? So then that is kept aside, and the other 2 are, one is acid and the other one is a distilled water. So acid also, it won't turn, the color will be same, red color only, and for the distilled water also color won't change. Now how we to identify whether it is acid or base? So just by smelling it, distilled water won't have any odor, so it is a odorless liquid. So and acid will have a that strong smell you can sense for the acids. So like that you can identify which one is acid, which one is base, and which one is the distilled water. Just if it is turned into blue, red litmus is turned into blue, it is base. So the other 2 are not turning the color, the same color, red color will be remained as it is. By smelling it, you can identify whether it is a acid or a distilled water solution. Here in this activity, to study the behavior of different acidic and base solutions using indicators. So what are the indicators that we have? Red litmus, blue litmus. We are given different types of indicators: red litmus, blue litmus solution, and phenolphthalein and methyl orange solution. These are given. So and we different materials are given here. So common laboratory acids are there. What are the common acids? HCl, hydrochloric acid, right? Sulfuric acid, nitric acid, and acetic acid, CH3COOH. These are the common laboratory acids. And coming to common laboratory bases, what are the bases? Sodium hydroxide, NaOH, calcium hydroxide, CaOH taken twice, potassium hydroxide, KOH, is it not? And magnesium hydroxide, MgOH taken twice, and ammonium hydroxide. These all are the common laboratory bases. And coming to the indicators, we are given different types of indicators here. So now what we have, tabulate your observation in the table. They are asking us to tabulate the observations. So how to proceed? Place a drop of each solution on separate watch glasses. So we take watch glass and we put 1 drop of that solution in the watch glass. Test the solution one by one using the indicators. Different watch glasses we have to use for different solutions, right? So first one, red litmus. So check whether it turns into blue. So first you keep all your materials here. What are the different materials? Basic acids, laboratory acids and bases, right? These first in acids also we are given 4 types: HCl, H2SO4, HNO3 and CH3COOH, right? 4 acids are given and 4 bases are also given: NaOH, CaOH, KOH, MgOH, like that. So now so if for first one, red litmus, if the red litmus turns into blue, then you have to check red is turning to blue or not, and we have to check blue is turning into red for each one. So if it is a phenolphthalein, observe the color changes in the acidic and basic media, how it is going to change the color. So now you need to take the records, right? So first one, if it is a acid solution, turn blue litmus to red. So a blue litmus solution, if you take, it will be turned into red using a acid solution, is it not? So for acids, right? For bases, keep the red litmus and red will be unchanged. For a acid solution, red will be unchanged and blue will be turned into red. So no color change and remains colorless. So this won't, no color change, right? And methyl orange solution, it turns into red for a acid. Coming to base, if it is a base, red will turn into blue, right? And blue will be unchanged, and phenolphthalein turns pink if it is a base solution, and it turns into yellow. So methyl orange solution will turn into yellow if it is a base. Now one by one, if you take check HCl, first if you take HCl, so no color change you will observe with the red litmus. So blue litmus will turn into red, right? And phenolphthalein, no color change, and this will be methyl orange solution will turn into red, like that. So this type of observation you can do and you can prepare the chart. So basically what is the understanding here? So how the acids and bases react with different indicators, recognize the characteristic color changes for acids and bases. So these principles we apply, and we try to identify whether it is a acid or base using these indicator tests. In this activity 2.2, so basically to identify acids and bases using olfactory indicators. Here we are using vanilla essence, vanilla, onions and clove oil. These act as a olfactory indicators, so which change their smell in acidic and basic media. This is the activity. So here what are the materials that we are using? Cloth strips soaked with vanilla essence, onion extract or clove oil, anything you can take. So we need test tubes here. Test tubes we are taking. Take dilute hydrochloric acid solution, HCl, and dilute sodium hydroxide solution, NaOH solution. It is a base, it is acid. So water for rinsing the cloth strips. Cloth strips we are taking. So what we are doing, procedure, here it is. Take 2 cloth strips soaked with either vanilla essence, onion extract or clove oil. You can take any of these things. So vanilla essence or onion extract you can take, or clove oil also you can take. So now smell and record the order of these strips to establish the baseline. So first you keep them for overnight and you smell it, okay, before conducting your experiment. So now step 2, this is step 1, first soaking. So now rinse the one strip with water and check whether its order changes. Record the observation. So first we rinse it. Step 3, add few drops of dilute HCl, okay, to one test tube. That is in one test tube we are putting HCl and in the other test tube we are keeping NaOH. So soak one cloth strip in the HCl solution and the other in the NaOH solution, two solutions. So we smell the cloth strips and after soaking them in acid and base solutions. So we note the change in the smell of for each indicator. So our observations here you have to tell. So if it is a vanilla essence, acidic solution, if you are keeping in HCl, so smell of vanilla essence remains intact. Basic solution, smell of vanilla essence diminishes. So in the acid, the smell is existing. In the base solution, you cannot find that odor. So in even onion extract also, acidic solution, smell of onion remains intact. And in the base solution, that smell diminishes or disappears. Clove oil also, it is also having a very strong smell. So in the acidic solution, clove remains intact and base solution, smell of clove diminishes. So finally, what is the conclusion here? Substances like vanilla, onion or clove act as olfactory indicators, right? Because their smell changes in acidic and basic solution. Olfactory indicators means they are using the smell, you are going to identify the type of material, whether it is acid or base. So in the acid medium, smell remains intact. In the base medium, smell diminishes or disappears. So this property can be used to differentiate acids and bases in the absence of visual indicators. These are the smell type of indicators. They are not the visual indicators. So previously we observed the visual indicators. In this case, we are taking, these are the using our smelling, we are going to indicate these materials, right? So